Welcome to mini lecture number 1 for module 5 on samples and sampling distributions. By the end of this module, you should be able to understand the ideas of random samples and random assignment and bias, which is the point of this mini lecture. In the next mini lecture, we'll look at how sample sizes affect the distribution of means, the central limit theorem and why it's important, and relate sample sizes to the values of standard error. And in the third mini lecture, we'll look at calculating and interpreting confidence intervals about sample means. So why is understanding sampling important? Well, the sampling designed for a study, if it's improper, can have an impact on the data collected and on the conclusions drawn. And often we strive for random sampling and random assignments to treatments. But if a study is poorly designed and has confounding effects or bias, the damage is already done. Nothing can be done to fix a poorly designed study. The only remedy is to repeat the study with the sound design, and so the best strategy is prevention. In this cartoon, of course, they have a random number generator, but it's not random at all. So random samples, when a sample is randomly selected from a population, it is called a random sample. And values in a random sample are actually representative of the distribution of values in the entire population because there's no bias. There's no bias because it's random. So how might we randomly sample a population? So here's an example of 30 individuals in a population. One thing that could be done is to assign each of those individuals a number 1 through 30, and then write those numbers down and put them in a hat, and then randomly take out numbers, and the first 10 numbers that you select get associated with the individuals, and those individuals would then be chosen. So if individual number 5 was picked out of the hat first, well, individual number 5 here highlighted in green becomes part of that sample, and do that 9 more times. Another way to do that with technology could be to have these individuals assign numbers, then write 1 to 30 in an Excel program, get it to randomize them, and then the top 10 numbers that after randomization are the individuals that are selected for the study. So in a random sample, each individual in the population has an equal chance of being chosen for the sample in simple random sampling. And the key points here are that random sampling helps to control systematic bias, but even with random sampling, there is still going to be sampling variability or sampling error, right? Because of those 30 individuals in that example, if we're only picking 10 of them, well, we pick them and we get an average of those 10 individuals. Well, if we do that again and we pick 10 individuals and they're a different set of 10 individuals, that's going to have a different value than our first sample that we did. And mini lecture number 2 is going to go more into detail about this idea of, with random sampling, looking at sampling variability. And if the statistic does not change very much, if you repeated this study over and over again, then you actually have very reliable information. So if there's not a lot of variability, it's actually quite precise, but that's going to depend on the original variability in the data as well. And how can you randomly assign individuals to treatments? So when we look at random assignment, that's now when you have individuals or subjects and you're going to put them into the different treatment groups. And it has to be due to chance in order for it to be random. So you could toss a coin, and if it's tails, they get assigned to one group, and if it's heads, they get assigned to a different group. Or using a random number table. And this type of approach helps to avoid these confounded effects and bias. Now, it's not always possible to randomly assign individuals to treatments, and many studies in medicine and healthcare are actually observational rather than experimental. And this is because studies comparing people with a disease to healthy people are necessarily observational. you can't take some individuals that are healthy and then assign some of those healthy individuals to have a disease treatment and then induce the disease in those people. And so many observational studies have actually provided very valuable information, and most human diseases could not be studied in other ways. And then instead of randomly assigning people to disease group, researchers obtain groups of people who already have disease and then have people who do not have a disease to make some comparisons. However, we have to be careful with observational studies because confounding factors can play a big role. So, for instance, if we're studying the effects of aerobic exercise on maternal self-esteem and physical discomfort during pregnancy, we're going to have two groups of women. So 31 women attending prenatal exercise classes and 22 women attending childbirth preparation classes. In this study of these two different groups of women, they found that the exercise group had higher self-esteem scores and lower discomfort scores, but we can't conclude that aerobic exercise results in higher self-esteem and lower discomfort, because there are other factors that might influence whether or not a woman is going to be attending prenatal exercise classes, for instance. And with this experiment, this study that was done, there are some major differences between these two groups socioeconomically, in terms of education, income, and occupation. And so this table shows that for the exercise group, only 19% of them had less than a college degree versus almost 59% in the non-exercise group. The exercise group had 19.4% of the women whose income was less than $30,000 per year versus the non-exercising group that 68% of the women made less than $30,000 a year. And then again, with unemployment, was also higher in the non-exercise group than in the exercise group. And so when we're thinking about maternal self-esteem, the effect of exercise is going to be confounded with all of these other factors that individuals are dealing with in their lives. So we have to be careful, again, with observational studies to ensure that this confounded nature doesn't happen. And it's important to be aware of it. So when we talk about bias, or accuracy, and error, or precision, we can think of it as a bullseye. So where the center, the yellow part here, is actually what the true population value is. And so if we have an estimate that's very accurate, it's going to be reflecting the true population value. So there's low bias because it's actually reflecting that value, even though there might be some variability. So the precision might vary a little bit. And if it's inaccurate, that means it's way off the bullseye. So that's the lower two panels. And that's showing that there's some bias, that for some reason it's moving to the upper right of these panels. And when it's biased, that means that we've over-selected or under-selected from certain groups in a population that will likely result in values that are not representative of the true population value. In order to correct these types of things, random sampling is always something to try to achieve, although is not always possible depending on what the study is. But remember that nothing can be done to fix a poor sampling design. And that concludes this mini lecture.